Então, dando seguimento ao nosso evento, eu vou chamar agora a professora doutora Ana Krook, também da Suécia. Ela vai falar sobre exercício físico e comunicação entre adipócitos e o sistema imune. A professora Ana é bacharel em Ciências Biológicas pela University of East Anglia, no Reino Unido. Ela é mestre em Ciências pelo Departamento de Patologia da Queen's University, no Canadá. Doutora em Filosofia pela School of Clinical Medicine na University of Cambridge, no Reino Unido também. E atualmente professora de Fisiologia Integrativa no Departamento de Fisiologia e Farmacologia e presidente do Programa de Pesquisa Estratégica em Diabetes no renomado Instituto Karolinska, na Suécia. Professora Ana, thank you for joining us today.
Thank you very much, Andressa. I don't think I've ever been introduced in Portuguese before. It was fun to hear.
Yeah, it's your first time.
Brazil.
Yeah, it's now time to hear from you. Feel free to begin whenever you are ready.
Okay, I will share my presentation here.
Yeah.
And I hope that you can see my presentation. Is that so?
Not yet.
Not yet. Okay, we will try again. See, we had a lot of time to practice this over the pandemic. Let me see again. Let's see. I want to share. There. Now I think it should be working better.
Yeah,
and we'll put it into presentation mode.
Yeah, you should share the window, the window.
Is it the other? Is it? I need to switch.
No, is everything okay? No. You can go.
Everything looking good?
Thank you. Yeah.
Okay. Well, so then thank you very much, Andressa, and thank you also for the invitation to speak here. And in contrast to my colleague, Dr. Duarte at Lund University in Sweden, I will not be able to take questions in Portuguese. But hopefully you will, we will be able to have a good discussion anyway. So I will speak to you today now for the next 40 minutes or so about our work that's focused very much on exercise adaptation and also in the context of immunometabolism. So I just wanted to share this really important slide first. I share my laboratory with Professor Jeline Zerath and Professor Harriet Wahlberg. And so we're quite a large group of people. We work at the Karolinska Institute in Stockholm. And our main focus is really type 2 diabetes. It's the disease that we work on from many different angles. And so hopefully I'll convince you that this is an important disease also with the immunometabolism. And the number one tissue that we focus on is skeletal muscle. And of course, Professor Duarte spoke about the brain. There are many important tissues in type 2 diabetes. It's a clearly multifactorial disease and many tissues interact. So why are we 30 people working intensely on skeletal muscle? Well, it goes back to some, just to remind many of you that you probably read about sort of in biochemistry and metabolism 101, and that we've known for over 30 years now that the main tissue that takes up glucose in response to insulin is skeletal muscle. So I'm sharing with you this picture where in green you have glucose uptake in people who with normal glucose tolerance or type 2 diabetes. And this was done basically by catheterizing across different tissue beds in control and population and type 2 diabetic population and measuring the uptake of glucose in response to insulin. So after a meal, for example. And as you can see, the yellow part is muscle, and muscle is clearly the tissue that is very much impaired in its response in people with type 2 diabetes. We also have adipose tissue, which is a very insulin responsive organ, and here we have the uptake across the splanchnic area. And one can, of course, think about here on the left now, I'm showing you just a schematic of a person. And of course, if we try to sort of estimate how much of a person is skeletal muscle, it's somewhere maybe between 55 to 35% of your body weight. So clearly it's a very large tissue, which probably explains part of the reason of all the glucose uptake. But the important thing is it's the tissue that's really impaired in the context of type 2 diabetes. So we're all focused on trying to understand why muscle insulin response is so impaired in people with type 2 diabetes. But I want to talk to you about exercise. And actually, Dr. Duarte mentioned towards the end of his lecture that exercise can very much be used in the context as a non-pharmacological means of treating metabolic disease. So why exercise? And indeed, I'm now again sharing with you some really old data, but I think it's really quite seminal and quite simple and just shows what the key sort of observation is. And so in this picture, on this particular open circles here shows an oral glucose tolerance test that's been done in 10 different men, 3 that had impaired glucose tolerance and 7 that had type 2 diabetes. And this is a pathological diabetic response to a glucose tolerance test. The plasma glucose goes up and then comes back down again. But you start at the type 2 diabetic level. Now, these men did 6 supervised exercise sessions. And you can see what they did. They basically did a treadmill or a cycle ergometer. And it was quite intense. But remember, we're talking about 6 supervised sessions and 1 unsupervised session. 1 week of exercise treatment. Now, this is in the black lines or black dots here is the response to the glucose challenge after that 1 week of exercise. And you can see that there is a remarkable improvement in these 10 men in 1 week. Now, despite the fact that it's now 2025, we have GLP-1 agonists, and we have the Ozempics, the Wegovys, we have no treatment that works as well as exercise. So there's an untapped potential to understand how we can treat metabolic disease. So what do we know about what happens in your muscle when you exercise? Well, just one single exercise bout. So if you were all now, after the lecture, deciding to just run for 20 minutes, you would have an increased muscle glucose uptake immediately in your muscle. And you have this increased energy demand. And what you have really is a disruption of homeostasis. And this disruption also improve. Not only do you increase your muscle glucose uptake, and that's independent of insulin. Insulin isn't important here, but afterwards you have enhanced insulin sensitivity. And this is something that we know. So if somebody is dependent on insulin as type 1 diabetes, they will have to change and reduce their insulin dose if they had exercised previously. And if you continue to exercise every day, you have exercise training, which is really the sum of many, many different exercise interventions, but you have muscle remodeling. And we all know, say we were to train for a marathon, we'd suddenly have enhanced endurance, we have more metabolic flexibility, and we have this maintained enhanced insulin sensitivity. So what we want to really understand in the lab is we have this increased insulin sensitivity, we also know increased training, mitochondria, and the only input that we've done is training. So in a way, it's a beautiful system to study because you can study the same person before and after exercise training. You keeping your genetics exactly the same. You can do this in human patients, and we're trying to understand what is the input. And of course, we know that there's very dramatic remodeling in response to exercise. And there are numbers of things that can happen. And we have been studying different transcription factors, metabolites, epigenetic events such as histone modifications and DNA methylation, and non-coding RNA. But the first sort of vignette I want to share with you, is really looking at the transcriptomic profiling. And this is some work in our research group that's been headed by Nicolas Pillon, an early stage researcher. And what Nico did a couple of years ago now is he mined my apologies. He basically mined the data that was out there in PubMed and looked at all the downloaded from more than 100 study groups, as you had more than 700 people, all the different studies from exercise interventions in humans. And then what he did was he filtered this. So you see that whether they were acute aerobic exercise, acute resistance exercise, or longest term training, or training resistance. And he also compared to inactivity. And so, as I said, this was a number of different groups, a number of different volunteers. He's updated that since this has been published. So there's now more studies that have been included. And this is all available at this website, metamex.eu. And it has an interface so that if you have a gene that you're really interested in, you can just go to this website and you can study this gene. And it'll tell you how it will basically behave in all these different exercise interventions. But just to share with you some of the overview and what we learned from looking at this. So this is some PC principal component analysis plots. And what I want to draw your attention to and what sort of surprised us, we know that if we do resistance training, so if we lift weight, we have a very different response in the muscle than if we were to train for a marathon. What we found in the gene response to exercise is that what really differed was here we have the acute response to an exercise intervention, and that's both resistance and aerobic. And on this side, we can see the training response. So either training for a marathon or training for weightlifting, but they both end up on this side. And a bit more difficult to see is we also have the inactivity response, which I think also tells us it's not always the opposite of exercise, but it tells us how muscle remodels in response to not being used. What Nico could show then is that there was quite different signatures with inactivity, the training, many exercise bouts, or the acute exercise bouts. And here we have some of the key genes that were clearly important for this. I want to draw your attention to one of the ones that we were really interested in. This is acute response of NR4A3, also called NOR1, which actually is shared across these two sites. And even though it's not seen here, it's actually something that's upregulated with exercise and downregulated with inactivity. And so we've been following that up in the lab. And this basically shows you, if you go into the MetaMex database, you can see the response. You can then select all the different studies, and this you can then basically pull out and see what happens to your gene of interest. NR4A3 is very responsive to inactivity. So you can see it goes down with inactivity, but it's also incredibly exercise responsive. And it's a transcription factor. And we think that it's a key regulator of the response to an exercise intervention. And basically, we think it's one of the central regulators of actually maintaining skeletal muscle mass, rather than perhaps so much regulating metabolism. Now, one of the things that the MetaMex database allows us to do is to really dial in on the time aspect, because we realize, of course, that when you exercise, you will have different waves of genes being activated. And if you collect enough of the data, so what Nico did is he basically went to see, well, which genes are activated immediately after, just within an hour after exercise, and which genes are activated 2 days after the exercise intervention. And we saw some quite interesting patterns. And what we can see is that a really key response to an exercise, sort of a reasonably hard exercise training bout, is this cytokine-mediated signaling. cytokine production, and general sort of inflammatory markers that we all know are important for muscle remodeling and important for a healthy response to exercise training, or here's just one exercise bout. And by following these different waves, you can also glean sort of how the muscle is set to remodel in order to change its metabolism. And here are some other genes that are more responsive towards like a day afterwards. And now you can see that the mitochondria are beginning to remodel, and we have these more metabolic effects. So the first wave seems to be an immunological wave, and the second wave of transcripts and modeling really seem to be focused on metabolism. And then one can look at the different forms of exercise or inactivity here have different impact on different kinds of genes. So we can see that the acute response or the training response really affects the ability of the muscle to handle lipids, and that is something that seems to be downregulated with inactivity. So here we can sort of see that inactivity is the opposite of exercise. The same thing, we can see that aerobic training is really the thing that increases your mitochondrial respiratory chains, and inactivity goes the opposite direction. And when it comes to the immunological remodeling, it seems that the acute aerobic is what really seems to have the biggest impact on this. So that's giving you a little bit of a background on how we've been thinking about this project and using this. But so then the question is, does everyone respond the same? And I guess to a certain extent, we all know that that's clearly not true. Not everybody responds the same to anything, really. But is there some sort of more, can we categorize people in different ways? And so we looked at the number of people in the Metamix database who were classified as being healthy or as being metabolically impaired. And this is a combination of people who are very obese or who were sort of classified as having diabetes. And we can see that there's, when it comes to the aerobic response, it's a little bit blunted, even though the fold is pretty much similar. It's a bit blunted in the people who are metabolically impaired, which made us sort of think that maybe the response is more altered in people who have type 2 diabetes. So I guess I just included this particular slide here really just to remind us all of the very complicated response to how muscle needs to adapt to higher intensity exercise. And just to remind you that really muscle goes from perhaps being completely inert and sedentary to performing explosive work. It really does disrupt homeostasis, and that muscle has an exquisite system to change how it responds to the demands that we place on it. So I'd like to now share with you some of our more recent data and look at many different ways of looking at one single exercise bout and tell you three things about sort of vignettes. One about muscle exercise-induced changes in mRNA in people with normal glucose tolerance or type 2 diabetes. And then, as was in the title, really talk a little bit more about the adipose tissue crosstalk and how that works in exercise, and compare people who have normal glucose tolerance or people who have type 2 diabetes, and then finish off with some unpublished work really on exercise-induced changes in metabolomics. So let me first introduce to you the setup. So we're studying a group of people who are age and BMI matched, and men and women who have either normal glucose tolerance or type 2 diabetes. And so they come in to our clinic. We take a muscle biopsy from them, and that's what we call the basal. They then get on a bike and they cycle for 30 minutes at 80%, 85% of their maximal heart rate. It's pretty intense, pretty hard, but they could all do it. And so it's adapted to each person. So it's each person's 85%. And they do this for 30 minutes. They get off the bike and we take another biopsy. We take a blood sample and we take the muscle biopsy, and just next to the muscle, on top of the muscle, we also take a fat biopsy from these people. And then we've done a number of different omic analysis. And so we look, and then we allow them to rest for 3 hours. They don't get to eat anything, and they just rest for 3 hours, and we take a third biopsy. So then we can compare the basal to immediately after, when they get off the bike, or 3 hours later. And the reason we pick the 3 hours later is that when we want to look at things like mRNA or proteins, it sometimes takes a little bit of time for the response to happen. So let's see what happened to this exercise-induced change in transcriptomics. So again, this was headed by my colleague Nicolas Pilon. And so we have the 3 sessions. And here actually I'm only going to show you data from the men, because when we did the study, we'd only recruited the men that were in this particular study. So this is what happens in the muscle. So here I'm showing you now volcano plots. So here's the normal glucose tolerant people, and the post means just when they got off the exercise. So 30 minutes after exercise, we have these genes that change significantly in the muscle. And if we just go down here, now this is the people with type 2 diabetes. We have also the volcano plot with a number of people. More genes go up than go down. And the groups, if you just look at them, look reasonably similar. 3 hours later, just like I said, a lot more things have happened. So we have a lot more genes that have changed. And it's very hard to see here, but the really interesting thing is that here we have about 500 genes that have changed in the recovery in the normal glucose tolerant people. But in the people with type 2 diabetes, we have 4,400 genes that have changed. So there's an order of magnitude difference in the number of genes that changed. So that actually surprised us a little bit. And so we were thinking a long time about what is it about type 2 diabetes that makes you change so many genes? And so what we think is happening, and the title says here that it's an exercise-induced infiltration of skeletal muscle by immune cells. We don't actually know that it is infiltration, but what we know is that the signaling pathways that are really different in the people with type 2 diabetes are signaling pathways and genes that really give you an immune cell signature. So when you exercise and you're a person with type 2 diabetes, you induce genes in your muscle or in your muscle tissue. You have to remember it's a whole tissue that really have a dramatically different kind of signature than in normal glucose tolerant people. We tried to visualize this. Now, in contrast to animals, in humans, the inflammatory response is not as pronounced. And so, but we can see a few immune cells. So here's basically, I'm hoping it's projecting well. Here you have a cross-section of muscle, and we've stained with CD11B plus for sort of a general immune cell marker. And this is what a normal glucose tolerant muscle looks like. And here there's a few more bright cells that are lit up in type 2 diabetes. But hopefully what you can see that even in the normal glucose tolerance, there's more, a little bit sort of in this 3 hours after they get off the bike, but there's more of this evident in the people with type 2 diabetes. So we do think that it's either an activation of the immune cells, or for something, there's a signal that comes from the muscle that somehow calls out, and these cells infiltrate the muscle in people with type 2 diabetes. Now, another way of looking at this, and this is something that we then remember that, many years ago, over 10 years ago, we'd already been studying to see that there was this hint of the people with type 2 diabetes have an increased inflammatory tone, even at rest. So even when we couldn't actually detect the immune cells with antibodies, we can detect this sort of activation of STAT3, indicating that there's some kind of increased immune cell activity or immune activity going on, which could also be in the muscle itself, which is always difficult to know when you're sort of dealing with a whole tissue. So we then sort of decided to have a look at the adipose tissue. That was so it's exactly the same setup. We're now also, and this was headed by Lucille Dolay in the lab, but we're now also including women with or without type 2 diabetes, and now focusing very much on the adipose tissue. So the interesting, or we were perhaps not so surprised, but here again, I'm now showing you this in a slightly different way. Here are the genes that changed when they got off at 30 minutes after the exercise, and there's a reasonably good overlap. There's a few more genes that change in the people with type 2 diabetes. Here we have, and then in the people with normal glucose tolerance. So this is the adipose tissue just next to the muscle. But again, we saw this very, very different response at 3 hours. And again, a lot more genes. So where we have maybe 1,300 genes in normal glucose tolerance, we have over 4,000 genes changing in the people with type 2 diabetes. And just to remind you, this is very similar to what we saw in the muscle. So something is really quite different in the people with type 2 diabetes. Now, back to the adipose tissue. And so one of the things that we were sort of interested in here now is thinking that adipose tissue responds to exercise. That is, of course, interesting, but we thought that perhaps it's also signaling in some way to the rest of the body. And so we were interested in these little pizza pies that you see here, is the proportion of the transcripts that would be predicted to be secreted. And that proportion actually is reasonably the same. So even though here we have 1,300 transcripts, about maybe a bit less than 25% are secreted. And it's the same with people with type 2 diabetes. But of course, now we have 4,300 transcripts to select from. But we were sort of interested in trying to understand, are there changes in adipose tissue secreted proteins that happen in response to an exercise bout? And so those were the genes that Lucille and I were interested in looking more at. But I would also just like to say to all of you who are interested in these sort of things is that we have deposited all the mRNA transcriptomic sequencing. So these genes are, of course, there for you to mine and think about things that you might be interested in. But we pulled out these ones. And so if we look at the sort of secreted genes or exercise-induced secreted genes in adipose tissue, there are a number of genes that have been previously reported as IL-6, which is clearly an exercise responsive gene coming both from muscle and adipose tissue, and a few other genes that are all increased in response to exercise. But we wanted to study these four we were more interested in because they hadn't really been reported so much. And one that really caught our attention was this oncostatin M. And here, the blue dots are people with normal glucose tolerance, and the red dots are people with type 2 diabetes. And you can see there's this peak that comes up immediately. So this is immediately when they get off the bike. We can see that this peaks very much in the transcriptome. We also looked at GDF15, SFRP4, and MXRA5 that also are responding to exercise in the adipose tissue. Now, why did we get interested in oncostatin M? Well, it's a cytokine that's been studied previously with potentially beneficial metabolic effects. And there'd been a study a few years earlier looking at diet-induced obese or OB mice. And when they were treated with oncostatin M, the response was a reduced obesity, they had less adipose tissue inflammation, and less insulin resistance. So we thought that was quite interesting. And in mice, the same group showed that exercise increased oncostatin M very specifically in the skeletal muscle fibers. But we now had found it in the adipose tissue of the humans, but we thought this would really be quite an interesting angle to pursue anyway. So we then also had the plasma from the same people. because, of course, in the adipose tissue, all I showed you was the mRNA. Now, in the plasma, we can measure the actual circulating oncostatin, and as you can see, it does increase progressively in response to exercise, and more so in people with normal glucose tolerance than in type 2 diabetes. And we could also, now we have both men and women, we can actually see that there's a little bit of a sex difference, not so much in the women, but the difference between the type 2 diabetics and the normal glucose tolerance is really driven by the men with type 2 diabetes that have a lot less circulating plasma oncostatin. And of course, here now we don't know whether it comes from the adipose tissue, but adipose tissue is definitely a potential tissue contributing. So what does oncostatin do? Now, oncostatin M will, if you study it in vitro, so we used human in vitro differentiated adipocytes, and we used all these 4 different circulating proteins, and we treated the human differentiated adipocyte, and what we found is that oncostatin M very potently increased phospho-ERK. And then we went to look at our biopsies from the people, and we could see that specifically both in the normal glucose tolerant and the people with type 2 diabetes, at the time when we see the peak in oncostatin from the adipose tissue transcriptome, we at the same time see a peak in increase of ERK. This, of course, is correlative because it's very hard to do something very specific in the humans, but at least it was in agreement with that oncostatin could be signaling to activate ERK, for example, in the adipose tissue. Now, we were also intrigued to figure out which part of adipose tissue is oncostatin M really coming from. And so we basically used some single cell data, which where we looked at the oncostatin M itself, so the secreted protein that we were interested in. And here you can see the adipocyte. So the adipocyte itself does not express very much oncostatin M, but if we go to all the different macrophages and T cells, there's a lot more oncostatin M that's expressed. When it comes to the oncostatin M receptor, or GP130, which couples with the receptor, you can see the inverse. You can see that there's a lot expressed in the adipose tissue, but not very much at all. So we hypothesize that if oncostatin M is primarily expressed in the immune cells, but the receptors, oncostatin M, GP1, and the LIF receptor, they are specifically enriched in adipocytes. So one could imagine that in response to exercise, we have this increase in expression coming from perhaps the immune cells, and that it signals to the adipose tissue. That is our hypothesis. How can we prove this in humans? It's always a little bit difficult, but we went on to look at, again, using the leg adipose tissue. We could see that we could blot for CD68 and oncostatin M, and this is using RNA fish. And you can see that there's a pretty good co-localization between the monocytes and macrophages, and we think that's the likely source of oncostatin M in adipose tissue. And this is both in normal glucose tolerance and people with type 2 diabetes. The other thing that's really different between humans and mice is the source. A few slides ago, I mentioned that previously it had been shown that in response to exercise in a mouse, it's the muscle that will secrete oncostatin M. We looked then in our humans, because we had the muscle tissue. Now, here is the basic detection limit of where you would say that that any gene is really expressed. And you can see oncostatin M, the count per minute are under, about 3 counts per minute in a transcriptome. So basically, human skeletal muscle tissue does not express oncostatin M. But it does express the receptor. Just to call your attention here, the top is 30, here the top is 2000. So the receptor is definitely expressed in muscle, but in humans, it seems that adipose tissue in response to exercise is probably what's secreting oncostatin M. So we think that this is our proposed idea of the intertissue communication. Probably when you're performing exercise, we're assuming that the initial signal probably comes from the muscle, and the muscle somehow recruits inflammatory cells, both to the muscle, but also to the adipose tissue. And the adipose tissue is responding with signals that then alter the metabolism. And so we thought a lot about how can we test this? And so we tried to test this in vitro. And bear with me now, I'll try and explain how we did this. So we took human THP1 cells, which as a sort of a model of immune cells, and we then treated them with something that we thought would be an exercise cocktail, because how can we have these cells cultured? So we treated the THP1 cells with cambuterol or forskolin, and then we took the medium. We basically measured in the THP1 cells whether oncostatin M, GDF or GDF15 were increased. And basically we could see that they were increased in response to this. And we could also see, because when we differentiate the THP1 cells into macrophage-like M1 or M2, we can see that in response to forskolin, the macrophage, sort of the M0s, the M1s, but the M2s are the ones that respond the most to make oncostatin into the media. So this is mRNA, and this is oncostatin secreted in the media of these THP1 cells. The other thing we did was we took THP1 cells, and instead of treating them with forskolin, we added media from cultured human muscle cells that we had contracted in vitro. So it's a bit of an artificial experiment, but again, we took this conditioned media from either people with normal glucose tolerance or type 2 diabetes. We had the cultures, we put this on our THP1 cells, and we could see that there was this wave of oncostatin M at pretty much the same time frame as we'd seen before, at 30 minutes, and GDF15, we had a longer kind of time frame. So we feel at least that it's possible that these different cells are talking to each other. And we're trying to mimic in vitro what we think is happening in vivo during the exercise. So just to summarize that, we think that the exercise induces inflammation in the muscle and the adipose tissue, and this exercise-induced secreted factors from muscle may contribute to macrophage activation and secretion of different metabolically important factors. So now for the last few minutes, I'll just tell you a little bit about metabolomics in the same group of people with normal glucose tolerance or type 2 diabetes. And this is just very much under work that we're doing at the moment. And so it's the same setup again, but now we're looking more at the metabolomic response. And so what I'm showing you here is if we just look at the metabolome in the muscle, in females with normal glucose tolerance, we have a lot of changes in branched chain amino acids at the 30 minutes time point, and that's quite a quick peak. And that's been known for a long time. We expect branched chain amino acids to change. And this is very similar in the men. In the men, we see also a more clear, and this is now in normal glucose tolerance, we also see that they have a change in fatty acids. Now, in the women with type 2 diabetes, we have quite a similar response again, but branched chain amino acids. And in contrast to the women with normal glucose tolerance, we have changes in fatty acids, long chains, polyunsaturated fatty, and some sphingomyelins that are popping up. And then in the men with type 2 diabetes, the branched chain amino acids are sort of like the key response, but we see some changes in lysophospholipids, and now DAGs as well are popping up as in response to exercise as a metabolomic change in the muscle tissue. So try and just summarize that a little bit more for you. So branched chain amino acids are clearly similar in all the responses, the TCA cycle and most acyl carnitins. Where we see more disease-related difference are in glycolysis and in diacylglycerols. And diacylglycerols have long been thought to be maybe playing a key part in insulin resistance in muscle in type 2 diabetes. And sex-related differences we also saw, and these were, again, the long-chain PUFAs were pretty much expected to see, but also in lysophospholipids and sphingomyelins that we don't think have been studied as much before. And now for just the sort of the last concept I'd like to share with you is not so much, what changes, but the timing. So here I've tried to illustrate a wave of that. When do the amino acids peak between the start of the cycle and just getting off, or the start of the cycle and the 3 hours later? And so here you can see sort of the wave of response in normal glucose tolerant women. So first it goes up, then it goes down, and then it stays the same. That's how you can read this. So there's this wave of amino acids in normal glucose tolerant women. And then we have a reasonably similar wave in the men with normal glucose tolerance. So not a huge sex difference. But what we find now is that when we look at the women with type 2 diabetes, we find that the lipid response is very much delayed. And especially in the men, we find that the response is sort of moved over to the right side of this graph. And so if you think about that in people with normal glucose tolerance, if you just measure glucose, you will, in response to a glucose challenge, you have a sharp up and then it comes down. What we're seeing now, and now it's in response to exercise, is that what's a quick up and down in normal glucose tolerance seems to take a much longer time in people with type 2 diabetes. It's almost like when we change homeostasis, and now we're changing homeostasis with exercise, in people with metabolic disease, it takes a longer time to get back to homeostasis. That's a concept that we're just thinking a lot about at the moment. This is a picture I've borrowed from my close colleague, Julene Zerath, when she just recently gave a talk about the importance of physical activity and the fact that we shouldn't, when we talk about physical activity, sometimes we need to talk about physical inactivity. And a lot of modern behavior is perhaps really unphysiological, and physical inactivity and sedentary behavior is something of its own that's really increasing, predisposing to even like changes in brain function, as Dr. Duarte spoke just before me, liver function, muscle function, and of course heart and cardiovascular disease. Just a little plug there. But I'm coming to the end, so I'd just like to summarize what I've been trying to say, is that one summary statement is an exercise bout in a person with type 2 diabetes, it results in a very different response than the same bout in a person with normal glucose tolerance. And so when you stress the system, it really unmasks differences that you don't see at homeostasis or rest. And one of the key differences appears to be hinged around immunometabolism. But I think what the key questions that we now have on the horizon is that, we know that exercise is important for managing and preventing type 2 diabetes, but we need to understand how the exercise really works and what is the nature of the more altered immunometabolic response in people with type 2 diabetes. And I think a really important thing is that it may be that this is an adaptive response, and maybe that it should happen in people with type 2 diabetes, but we don't know whether it's a good or a bad thing at the moment. And I think mapping the molecular effects of exercise with advanced omic can really advance our understanding of muscle function and metabolism. And with that, again, I'd like to thank this fantastic group of people. Here you can see what everybody does when they have some time off. We go and do things like VO2 max tests and make sure everything is working in our exercise lab. And just back again to this group and to thank all my colleagues. And with that, I hope that it's been reasonably clear, and I'm happy to take any questions.